Okay, so hi everyone, my name is Valerie, and just before I start, I want to give a huge shout out to my mentor, Rabia and June, who provided me lots of helpful advice and a lot of guidance. So now on to my research projects. My research project basically aimed to develop a cost-efficient, user-friendly, and time-efficient way to observe long-term potentiation. The short form is LTP in hippocampal neurons. And it's specifically focused on analyzing LTP in images of neurons, just because there's a lot of ways actually to analyze LTP. So I analyzed LTP using neurons. And I did this by coding an analysis pipeline. So I just threw a lot of terms at you. If you didn't understand anything, please don't worry. I will be explaining everything in my presentation. So let's move on. First, I'd like to just start off with a few preliminary definitions. So first of all, the hippocampus is the part of the brain involved in memory and function. As you guys can see here, it's like this light blue kind of highlighted part in the brain. And second of all, neurons are the main cells found in the brain. Synapses are areas where basically the presynaptic and postsynaptic neurons send signal to each other, and this can either be electrically or chemically. And here's like a picture for reference. So this is the presynaptic neuron. The presynaptic neuron basically is the neuron sending the signal, and the postsynaptic neuron, which is over here, is the neuron receiving the signal. Now I just want to go quickly over what LTP is. So as I mentioned earlier, LTP is short for long-term potentiation. And in a nutshell, just because it's very complex, and obviously I don't want to get too into it, in a nutshell, LTP is the strengthening of synapses. And this is due to frequent stimulation through a synapse. And most common type of LTP is CA2 plus LTP. And it's basically when the postsynaptic neuron becomes much more sensitive to a neurotransmitter called glutamate. And I'd say like LTP, like while doing research, LTP is pretty important because it's thought to play a major role in forming memories and in learning in general. So I also want to talk about LTD, which is long-term depression, and it's basically the opposite of LTP. So LTD is basically the weakening of synapses, and the synapse becomes, is the weakening of the postsynaptic neuron's sensitivity to the neurotransmitter glutamate. So yeah, it becomes more weaker to glutamate. So now I'm just gonna go over some methods of analysis, both in neuroscience in general, and just maybe it's like how LTP might be analyzed. So the first method is called patch clamping, and patch clamping is usually used to determine the flow of ions in a single channel or a single ion channel on a cell membrane. Voltage clamping it's kind of similar to patch clamping, but instead of measuring the flow of ions in a single channel, it actually measures the flow of ions across a cell membrane. And finally, we have immunohistochemistry, which is the use of antibodies, which will usually contain like some sort of fluorescence, and they react with their opposing antigen to create a chemical reaction, which basically creates a stain. And this method, immunohistochemistry, is usually used for the identification of neurochemical molecules in neuroscience generally. So now that I've just defined a few basic terms, I'd like to go into my project overview now. My project focused, as I said earlier, on the analysis of LTP and aims to try to figure out if a neuron is potentiated or not relative to other neurons. So that is my main research question here. And I sought to answer this question with the use of an application called VG-ImageJ, which is usually used in science for image analysis. And I also used images of neurons, as I mentioned before, and those were provided by my mentor. I just want to note that this coding analysis pipeline that I coded, it's specific only to the images that I had. there's obviously like more sophisticated analysis pipelines out there that are more general and can do like a lot more different things that my analysis pipeline could. But like for the needs of this project and for my needs, like it was completely fine what I had. So yeah, overall, I just wanted to create a program that was easy to use, that was accessible and inexpensive, and like to that point, just didn't require too many materials just because I was doing TSI online. So I knew I wouldn't have access to that many materials. And most importantly, I did want to make this program time efficient just because LTP is very tedious to analyze in ImageJ if you're just doing it manually. So here's basically an analysis overview. So what you would have to do manually in order to analyze LTP. So first of all, you open an image and it splits the image into 3 channels. I'll be going over this a bit later, but the 3 channels are the green, blue, and red channels. Basically, yeah, so this was done for all the neurons that were analyzed. The second step was thresholding the green channel to define the area you want to analyze. So this basically creates like an area of interest. This is what thresholding does. If you could see on this bottom left-hand corner, this is like the image before thresholding. So this is the green channel image before thresholding. And after thresholding, this is what it looks like. So thresholding basically just gets rid of any interference that you don't want to track throughout, it gets rid of any interference that you don't want in your final results. The third step is to measure the mean intensity of the blue channel and the quantity of puncta. So puncta are basically binary points that the program detects in an image. And the fourth step is kind of the same thing, but for the red channel. So in the fourth step, the mean intensity is measured and the quantity of puncta is measured for the red channel. Finally, we save the files in your desired folder. So just a note that I won't be talking too much about puncta quantity. It's just an additional feature that I thought might be useful. Okay, yes, and just some clarification on channel splitting. So channel splitting is basically where you take this original image and you split it into these 3 channels, and each of these 3 channels represents a different staining protocol. So the green channel, which we have over here, this gives the viewer a general outline of the neuron, and this channel detects GFP staining, which stands for green fluorescent protein. And yes, as I said before, it gives the viewer a general outline of of the neuron, basically. The second channel we'll be looking at is the blue channel. So the VGLUT1 channel, which is over here, this stains the presynaptic neuron. And finally, we have the red channel. So over here, this was obtained using the GLUA2 staining method, and this channel stains or detects rather the postsynaptic neuron. And just wanted to say that, like, as I mentioned earlier, after splitting the channels, the program would calculate the mean intensity for the VGLUT1 and GLUA2 channels, so the red and blue channels. And this was done for 49 images of neurons in total, 32 of which were potentiated and 17 which were part of the control or the non-potentiated group. So let's move on to our results now. So, okay, this is first the GLUA2 or the red channel results. So in general, once the mean intensities for all the neurons were collected, I split them into the respective groups. So like if they were potentiated or part of the control group. And I found the average intensity for each of the groups and their different channels. So let's go over the red channel first. So the mean intensity for the potentiated group was about 107.286 with a standard error of the mean of 22.214. And the mean value for the control group in this channel was 115.129 with a standard error of the mean of 11.278. And we go on to the VGLUT1 channel mean intensities. So the mean channel for the potentiated group was 57.615, and the standard error of the mean was calculated at 18.481. The mean intensity for the control group was 60.019 with a standard error of the mean of 23.067. And just want to discuss quickly some my discussion. So give you a summary of the discussion and some of the limitations that were in my project. So first of all, you might have noticed that the error rates are very high, like unusually high. And first of all, there were a lot of images to analyze. Like, I think with more images, I would have been able to make a more accurate analysis, maybe more accurate description, but it's okay, it's fine. Also, many of the neurons that I was analyzing, they had different areas. And of course, this kind of makes it hard to compare to each other, especially when talking about mean intensities and when using the mean intensity to track LTP. There was also something else that was surprising. It was that in both cases, the mean intensities for the control group was actually higher than the mean intensity for the potentiated group. And this was surprising just because I thought that the mean intensity for the potentiated group would be higher. And the reason that this might have, or we might not have gotten the answer we expected was because there was an uneven amount of neuron images for the control group when compared to the potentiated group. Because as if you remember earlier, I said, so out of the 49 images in total, 32 were actually potentiated and only 17 were part of the control or the non-potentiated group. Just wanted to say also that, like, as a limitation, this program should be used in conjunction with other analysis systems, just because, like, you can only get so accurate with a system that you automate. Like, it's obviously much better if you have the time and the patience to do all of this analysis manually. But of course, just because this analysis, especially using ImageJ, VG ImageJ, because it's very tedious, I just wanted to make a program that would reduce the amount of time needed to calculate mean intensity. Okay, so that's kind of the summary of my project. Now I'm just going to talk briefly about my experience. So obviously I wanted to do neuroscience just because I've always found the brain to be interesting. Like, it's very diverse, I think, in like all of its systems. It has like, it's just, yeah, very diverse in general. And I decided to do this project specifically, so coding an analysis pipeline, because I wanted to challenge myself with something that I wasn't too familiar with already. And I guess like my main challenge to this was, first of all, not knowing much about LTP or LTD. And obviously, like, you just kind of learn along the way, like what LTP and LTD are. And yeah, just by reading a lot of articles and things like that. I also had little experience in coding, which is why that was a challenge as well. The interface that I was using, so VG ImageJ, was also really new to me. So lots of experimentation while working was needed. For those of you who have some experience in like coding or computer science, I also ran into a ton of errors as well. So that's a given when, I guess, working with coding. So yeah, that's kind of my presentation. Thank you all so much for listening.